Okay, welcome to video 4. We're now moving on to the specific immune system. What I'd like you to do here is to, as usual, answer the questions before we begin. So pause now and see how you get on with answering those questions. Okay, welcome back. So what we're going to do here is not to delve into the specific functions of each cell. We're just going to have sort of a broader look at the specific immune system before we start looking at the cells specifically and what they do. So which cells are involved in the specific immune system? Your answer should be this. So lymphocytes broadly, but specifically T and B lymphocytes. Right, the more important question, how are these cells specific for a pathogen? Right, let's do some drawing here. Let's go with blue. Okay, so we've got several types of cells here. We've got, and we'll go into these in more detail, but let's just do these. So we've got potentially helper T cells. We've got the other type of T cell, cytotoxic T cells. Sorry. And then we have B cells. Again, I've just drawn a nucleus and no other organelles. So the three types of cells we're going to deal with in more detail in videos to come. But just to talk about these, so these are part of the specific immune system, and what that means is they have receptors on their surface. So these T helper cells have receptors. Cytotoxic T cells have receptors, so they're proteins, remember. And then B cells will actually have antibodies. And again, we'll deal with those in more detail in a further video. But they're proteins on their surface. Each of these cells will have a different receptor on their surface. Will express different receptors. And each of those receptors will be specific for only one antigen. They will only fit one antigen. That's what makes them specific. The receptors on their surface on each helper T cell, cytotoxic T cell, and the antibodies on B cells are specific for only one antigen. So that's what makes cells in the immune system specific. How does a non-specific immune system connect to the specific immune system? Okay, well, this is through phagocytes. So if you remember, let's go through a different color here. Phagocytes engulf pathogen. That's contained in a phagosome. Lysosomes fuse with the phagosome. They release lysozymes, these enzymes, into the phagosome that then hydrolyze or destroy the pathogen. So you have, let's just show that phagosome, and the pathogen has been hydrolyzed, so it's been digested basically into fragments. The next thing that phagocyte does is that it takes some of that hydrolyzed parts of the pathogen, so some of the what is then becomes really an antigen, moves it to its cell surface, and I'm depicting like a protein on the surface of the phagocyte there. And what it does is it presents some antigen from that pathogen on its surface. We then call that cell something different. It's still a phagocyte, but it's now doing another specific job, which means it is an antigen-presenting cell. It does as it says. It presents the antigen on its surface. It becomes an antigen-presenting cell. This is how the non-specific immune system connects to the specific immune system. Let's go back to what we were talking about. We're going to focus on helper T cells because this is the cell, and we'll talk about that in more detail in the next video, that binds to this antigen on the surface of the antigen-presenting cell. So there will be a helper T cell in the body that has a receptor that specifically fits that antigen. It's said there are 10 million different types of lymphocyte in the cell, in the body. The reason they're different is because they each have a different receptors on their surface. And so one of them will fit this antigen on the surface of the antigen-presenting cell. That's how, and let's just show that. So let's say that receptor fits exactly, it will bind. There is the connection between the specific immune system, the helper T cell here, and the antigen-presenting cell here. So that's where the connection is. And the next video will go on to describe exactly what happens to those helper T cells when they've bound to the antigen on the surface of the antigen-presenting cell.